This brief presentation provides a concise overview of the framework for developing recommended intakes of bioactive dietary substances. The work was supported by the Institute for the Advancement of Food and Nutrition Sciences Bioactive Committee. I am Barbara Lyle, a co-developer of this framework. The framework authors have a wide range of experience, including various roles in establishing how dietary recommendations are developed to translating evidence into quantified intake recommendations. If you would like to read the relevant disclosures for each author at the time of publication, please pause the presentation now. This framework uses the definition of bioactives from the National Institute of Health Office of Dietary Supplements, which states that they are constituents other than those needed to meet basic human nutritional needs that are responsible for changes in health status. Why is this framework needed? Quantifying intake recommendations for bioactives differs from historical nutrient recommendations, which typically have an established deficiency condition as the outcome from which to set a minimum amount needed. Dietary bioactives instead focus on ranges of intake for optimal health. The framework was intentionally designed to be flexible so that it can be adopted by credible guideline developers globally. A key point is that it applies to food forms only. Supplements of extracted or concentrated bioactives are explicitly excluded as there is substantial safety considerations for these forms that go beyond the scope of this framework, and the guideline developed from this framework is to be translated in the form of bioactive food forms. There are 4 steps in the framework as shown on this overview slide, with go, no-go decisions after each step in order to determine whether to make a recommendation, and if so, then specifically what will be recommended. A structured conclusion statement helps guideline developers communicate the recommendation in the full context of relevant information, including the quality of evidence for the specific bioactive and health outcome, whether it's to maintain normal structures and functions in the body or to reduce the risk of specific health outcomes, and also to clearly state if the recommendation applies to the general population or to specific subpopulations. So let's look now very briefly at each step. The first step is to characterize the bioactive, the amounts in specific food sources, and quantify intakes. Food composition data are necessary in order to translate a quantified intake recommendation into food sources. Refer to the framework paper for details on how to answer all 3 of these questions in order to determine whether evidence is sufficient to proceed to Step 2. Step 2 determines whether there is a history of safe consumption so that the final recommended range is both efficacious and safe. Since this framework addresses food forms, these dietary bioactives typically have a history of consumption to inform a decision about safety for a quantified amount. Note that evidence is needed to ascertain a quantified level of dietary bioactive with no known health effects. A history of safe consumption applies existing principles already in use and offers flexibility by recognizing that specific approaches vary by the food regulatory practices in a specific geographic region. By definition, dietary bioactives are present in the diet and therefore have a history of use on which to base a safety assessment. The publication on the framework provides details on existing practices to determine history and level of safe use, but also when and how to address the exceptions that are noted on this slide, conditions in which experimental testing applies. This framework does not apply to bioactives consumed in supplement forms because isolated and concentrated forms differ in several aspects as listed on this slide, which need to be taken into account when evaluating safety. The purpose of the 3rd step, conducting the efficacy review, is to quantify the relationship between the specific dietary bioactive and accepted markers of health or normal function using state-of-the-art systematic evidence review methods. Notice that health outcomes can be maintaining normal body structures or functions. For example, for dietary lutein, the outcome could be to maintain normal macular pigment optical density in the retina at a level that maintains normal vision. Or the outcome could be related to maintaining health status, like maintaining normal blood glucose or blood pressure in the normal or optimal ranges. And finally, it could be a disease risk reduction outcome, such as reducing blood LDL cholesterol, which is a recognized risk factor for heart disease. The systematic evidence review is unique in that the relationship with the dietary bioactive must be quantified. For example, results expressed in the literature on reported studies in quartiles or quintiles of intake exposure need to be converted to milligrams or grams. If there is a moderate or strong evidence of relationship between the dietary bioactive and the health outcome, with quantities established, not just directional relationships, but actual quantities, then the final step can be advanced to the step of translating the association into a dietary intake recommendation. The final step incorporates the results of all three previous steps in order to first determine whether to make a recommendation, and if so, then translate it into a quantified bioactive recommended range from food sources. A few highlights. First, the framework establishes that moderate or strong quality efficacy evidence is needed to qualify for a recommended intake using a best practice method to grade evidence. Second, the framework sets the standard as a range of intake rather than a single threshold minimum. This is so that there is clarity on where the evidence is available on amounts that are efficacious, as well as with a history of safe use, reinforcing the concept that more is not always better. And third, the recommendation clearly states the quality of evidence, the health outcome, and whether it applies to the general population or specific subpopulations. To give you an example, the quality of evidence that's moderate or higher would be considered by the guideline development panel using the two additional criteria that are shown on this slide. If all criteria are met, the range of intake is then set. In the simplest scenario, the range recommended would be the same as the range demonstrated to have benefit with the specified health outcomes from the systematic evidence review. However, if the history of safe use shown on this slide as Y prime is below the range demonstrated to be efficacious, then the recommended range needs to be adjusted down to the level with established safe use. And finally, if the panel determines that a quantified intake recommendation will be made, the framework provides a structured format that clearly conveys the recommendation. A secondary benefit of using a structured statement is to harmonize between various organizations that may develop dietary bioactive recommendations. With funding provided by the IFN's Bioactive Committee, a flavanthriol recommendation has been developed that quantifies intakes for cardiometabolic health among the adult general population. First, a knowledge map was developed to inform the health outcomes for which evidence was sufficient to conduct a systematic review. Then a comprehensive systematic evidence review was completed that provided quantified amounts associated with the health outcomes, as well as it examined various food sources of flavanthriols. And finally, a guideline panel convened by the Academy of Nutrition and Dietetics translated this evidence, considered the history of safe use, and recommended a range of flavanthriols. It recommends that for the general adult population that they increase consumption of nutrient-dense foods rich in flavonthreols and low or absent in added sugars, including but not limited to tea, apples, berries, and cocoa. And based on moderate quality evidence, consumption of 400 to 600 milligrams daily of flavonthreols may reduce risk associated with cardiovascular disease and diabetes. In summary, this approach adapts standards of practice used in clinical medicine in order to quantify in a credible way dietary bioactive recommendations for foods consumed by the general public. If you are a credible guideline developer interested in applying this framework and would like to partner or simply learn more about the process, contact IFANS through the website listed on this slide. We look forward to future recommendations advancing quality guidance about dietary bioactives and health for the general population.